Hello, everyone. Welcome to today's desktop seminar by the Royal Society of Chemistry. This webinar series showcases leading research in topics across the chemical sciences. Today, we'll hear two excellent speakers discuss their work on the chemistry of inorganic clusters. We have Professor Stephen Ragsdale and Professor Patrick Holland joining us live. This webinar is scheduled for 1 hour. Each presenter will give a talk followed by an interactive question and answer session. At any time throughout the webinar, please submit questions using the Q&A function in Zoom, and we'll ask them after the presentation. First, some housekeeping. We must note that this webinar will be recorded and the recording will be made available afterwards. Because of this, you will all be muted throughout the event and I'll relay questions during the question and answer. Of course, we want everyone to be able to enjoy and contribute to the webinar, so we'd ask all participants to be respectful of one another and to follow the agreement on this slide. Before we start, I'd like to take a moment to highlight that RSC has a variety of journals relating to the inorganic and biochemistry like you'll see in today's webinar. Specifically, I want to shout out to Dalton Transactions as our home for inorganic research now and for more than 20 years. Last year, there were nearly 2,000 articles published for more than 3 million downloads of those articles. Plus some cool journal covers, and of course, we'd like to be able to add yours to the slide and hope that you'll consider Dalton Transactions for your next submission. Without further ado, I'd like to introduce our first speaker, Professor Stephen Ragsdale. He's the David Ballou Collegiate Professor in the Department of Biological Chemistry in the Medical School at the University of Michigan. He works on microbial biochemistry and has a focus on the role of metals in biological processes. In fact, he was the chair of last year's Metals in Biology GRC and has contributed to a textbook we published on the chemistry of nickel, which you can find at this QR code. Today, he'll be telling us about how nickel iron sulfur clusters are at the heart of enzymatic CO2 activation, reduction, and fixation. And now I'll hand off to Professor Ragsdale. Thank you.
You can see me fine?
Yes, looks great.
Okay. Let's see. Okay, well, thanks so much for the invitation, and I'm so happy to be here and talk about something that's near and dear to my own heart, which has to do with nickel iron sulfur clusters at the heart of enzymatic CO2 activation, reduction, and fixation. These are people in my laboratory and um, uh, collaborators who we've been working with for many years on this project. And I want to thank them for all their contributions and also the funding from NIH and Department of Energy for the work that I'll be talking about. And they've been funding us for many years, supporting this. So the outline, I want to talk in general quickly about giving you an intro to the organometallic biochemistry involved in the Wood-Youngdahl pathway of CO2 fixation. I'll talk a little bit about the catalytic mechanism of COD hydrogenase and work we've done related to that, and also the catalytic mechanism of acetyl-CoA synthase. So if we look at the heart of the Wood-Youngdahl pathway, we see that carbon monoxide dehydrogenase and there are metal clusters that are involved, shown here on the right-hand side. Let me just see if I can make this laser pointer. Something called the C cluster on COD hydrogenase. I'll talk more about that. It's a nickel iron sulfur cluster. And that takes 1 mole of carbon dioxide. Overall, we have 4 moles of hydrogen, 2 moles of CO2. They make acetic acid that comes in as acetyl-CoA, which is also fixed into cell carbon. The acetate is a product of this overall fermentation and it's thermodynamically favorable. So one of these CO2s comes in here, binds to the C cluster and makes carbon monoxide. The other CO2 comes in, makes formic acid, which then enters something called the folate pathway, where you can see it gets converted through organic type intermediates to make methyl tetrahydrofolate. A methyltransferase ends up binding that and transferring it to a coronoid protein. So that methyl group bound to cobalt is the first of the organometallic intermediates from this part of the pathway. And as I'll show you, there are organometallic intermediates over here on CODH as well. Okay, so that methyl group gets transferred off to this nickel, 2 nickel, 4 iron, 4 sulfur cluster called the A cluster on acetyl-CoA synthase. And that makes an intermediate that's bound to that complex, to that metal complex. That ends up making an acetyl intermediate when it binds carbon monoxide. Coenzyme A then comes in to make acetyl-CoA. So these are the organometallic intermediates in the pathway. The acetyl-CoA again goes off into cell carbon or makes acetate. So there are really some interesting characteristics about this pathway. Unlike any other CO2 fixation pathway, it's considered to be the most ancient CO2 fixation pathway and may have been the first metabolic pathway as life um, originated on Earth. As I said, it's based on organometallic chemistry with enzyme-bound nickel carbon and cobalt carbon bonds. The enzymes are highly sensitive, so this pathway is found only in anaerobic microbes, but it does constitute a lot of the CO2 fixation that takes place on Earth. It's the only CO2 fixation pathway that that can conserve energy. That is, it produces instead of requiring ATP like all other CO2 fixation pathways. Um, it allows microbes to grow on very simple gases, and right now this pathway is being used in the biotech industry to make chemical feedstocks like acetic acid, butane ethanol, and there are really a host of other compounds that these particular small molecules are being used. For example, even with Lululemon, this Lancetech is working with companies that are giving us a lot of different products from this renewable energy source, CO2. Okay, so it's a reversible pathway as well. Some organisms actually use this pathway for growth on acetyl-CoA, and some use it for acetyl-CoA synthesis. So because this is an anaerobic pathway, it's one of the things we have to do is make sure that everything is done anaerobically. So we grow cells, harvest them anaerobically. We maintain the proteins in anaerobic chambers at less than 1 ppm of oxygen. Um, Sometimes the enzymes require some sort of activation to get up into the range of activity we need for our spectroscopic studies. We purify and then concentrate these proteins, and this is showing one of the proteins we study at about 2 millimolar, which is required for um, some of the characterizations that we do. We also crystallize these proteins, and you can see beautiful crystals of different sizes depending upon the way that we make the crystals. That is, we use these dilutions of seed stocks and control the seed stocks to get the size crystals we need, whether we're going to be doing neutron diffraction studies, X-ray free electron laser studies, or cryocrystallography. So we determine our structures using XRD, as I said, XFEL, or serial crystallography. And also, these solutions are used for spectroscopy and kinetics, and I'll tell you some of the results of those studies. So I'll start with the catalytic mechanism of COD hydrogenase and talk about the metal clusters, the intermediates, the gas channels in this pathway. So there are different types of nickel COD hydrogenases. There are also a class of molybdenum CODHs I will not talk about today. But the CODHs shown at the top here are called standalone COD hydrogenases, whereas the one below also have another a protein, acetyl-CoA synthase, that's bound tightly to it in a complex, alpha 2, beta 2 from protein sort of nomenclature. The standalone CODHs generally have much higher activity and than the other enzymes. And I'm going to start talking about this first class of CODHs because if we're looking at the spectroscopy at nickel, it's important to have a protein that doesn't have two other additional nickels in your solution that are going to end up giving perhaps making it more complex to figure out what the nickel ions are doing on either protein. So in both cases, we've been studying the isolated proteins, in some cases by recombinant methods. So Some of the interesting things about CODH is that unlike most inorganic catalysts, the nickel CODHs are highly specific for carbon dioxide. That is, they don't reduce protons, even though proton reduction is much more thermodynamically favorable than CO2 reduction. They also catalyze CO2 reduction right at the CO2 CO couple. That is, there's no overpotential required, whereas most catalysts that are used in inorganic systems require an overpotential as high as a volt, in addition to the potential required for the actual catalysis. So this is a sort of skeleton picture of that CODH where you can see that there are proton and water channels, and there are three metal clusters here that allow it to catalyze the proton coupled electron transfer to CO2 to make carbon monoxide. And that occurs at a redox potential of minus 520 millivolts, again, without an overpotential, with very high catalytic efficiency and rate. So the C cluster shown here is a nickel iron sulfur cluster that catalyzes CO2 oxidation and CO2 reduction. It's a reversible reaction. Um, B cluster that's involved in electron transfer, and another D cluster that's sitting right at the interface of the two subunits and sitting also right at the surface of the protein. Now, the redox pathway is shown here, where you have the catalytic mechanism below, and you can see that all of these redox centers are very close to each other. And that's really important because the redox potential gradients between these different couples, as well as this very, very small distances, um, ensure rapid intra and inter-electron transfer from electron donors to acceptors without an overpotential. So if you look at the overall mechanism, which is diagrammed here, we have something I'm just called CRED2. We've been working hard for many years to try to differentiate between um, these two states of the C cluster that's shown here, drawn out as sort of as a Lewis acid or like a chemdraw picture, where you see the nickel ion, the 4 iron 4 sulfur cluster, shown here in CRED1, CRED2 without the water. But what I'm showing this is 2 electrons more reduced than this state. So this can be reduced by dithionite, many different chemical reductants, but that's also key to the whole process of CO2 reduction to CO, which of course is a 2 electron redox reaction. So the way we look at it is the protein, if we're going to catalyze CO oxidation. CRED1 state is where we would begin this reaction with this iron 2 state, a nickel 2 state. That ends up binding to carbon monoxide, forming a complex. We are right now trying to trap this complex under catalytically relevant conditions. And then that undergoes an attack by this water. There's a deprotonation that takes place. That water binds the CO2, the carbon monoxide, to make a carboxylate and a bridged CO that then an intermediate that is seen as shown here on the right. I wish I could get this off. I can hide this there. Okay, through this carboxylate type intermediate, and then that bridge forms to complete the catalytic cycle, release CO2. And again, this whole thing is taking place at 40,000 per second. So it's very hard to trap these intermediates because that's so fast. We're talking about microsecond, low microsecond time scales to try to trap anything. So what we've been doing is working with variants. I seem to be stuck here. To move this thing forward. Okay. Anyway, there we go. So, so our aims are to recombinantly express this protein with full occupancy of metal centers, full activity of the native protein, to generate variants with full occupancy, but hindered rates of CO oxidation activity. And that's really important because our rates again are too fast, and we want to be able to capture the intermediates, meaning that we want to have time to be able to mix the samples, the protein with its substrates. And then anaerobically spectroscopically characterize the catalytically relevant bound states of CODH both in crystallo and in solution. And then use spectroscopic and kinetic methods to validate these intermediates. So there have been some intermediates that have been shown before, the CO bound intermediate, the CO2 bound intermediates, as well as the inhibitor cyanide, which is competitive with CO. So these have been characterized, but again, they were not done under strictly anaerobic conditions, and they were not done under conditions that would allow one to trap intermediates at catalytically relevant rates. So um, the mutation the mutagenesis technology that's been taking place has been done by Kareem Abulson, who's been doing a lot of spectroscopy crystallography to characterize this system. And we do have a variant that I'll show you here, where you take at room temperature, catalytic rate constant of 971 goes down to 0.061 with this variant, and we still have high degrees um, of occupancy of our metal center in this sample. And so there's a proton channel that's linked to this. So what we've been doing is mutating residues that are in the outer sphere and things that we can be absolutely sure by crystallography. Of course, the 70% was determined by crystallography, and we're getting around 1.2 to 1.5 angstrom resolution on the crystals. So we still have unresolved issues that we are working on. What is the difference between CRED1 and CRED2? I think we resolved that recently in collaboration with Riti Mukti at SSRL, and structure of the CO intermediate. Also to try to determine the proton coupled electron transfer mechanism. So I wanted to just also now switch gears and talk a little bit about the catalytic mechanism of acetyl-CoA synthase. And the main things that we've been working on, uh, determining the structure and function of the nickel iron sulfur cluster in this active site, trapping and characterizing what was proposed to be nickel-based organometallic intermediates many years ago, and identifying a crucial alcove that surprisingly and interestingly is integrated to the carbon monoxide channel. So when we put acetyl-CoA synthase with CO dehydrogenase shown here in the middle, the structures of the COD8 standalones is um, almost identical to that of the same molecule found here bound to acetyl-CoA synthase. So the acetyl-CoA synthase has this A cluster, and that is involved in the conversion of CO and the methyl group with CoA to make acetyl-CoA. So interestingly, there is a tunnel that we have characterized. Kathy Drennan determined it by crystallography. We had done spectroscopic studies indicating this was highly tightly coupled. And you can see that CO generated at the C cluster actually travels through this. And this was done at high pressure xenon. So each one of these represent a xenon molecule, which is about the size of CO, and identifies where this channel is. And you can see that that goes throughout the protein structure. And we've done computational studies with Simone Roger um, at, um, at PNNL, who tells us that it's just hard to tell where the COs actually are. They're just zipping around through this tunnel very fast. But that pretty much follows the same path here. It's just now we know more about the dynamics of that. That's unpublished at this point. So if I just look at the acetyl-CoA synthase mechanism, I've shown you CODH. I've shown you the gas tunnel just a second ago. That binds to make a nickel carbonyl intermediate. A methyl group can then bind to that, make a methyl bound intermediate we have not yet trapped. That methyl group is donated by the folate pathway, as I said earlier. This actually is a bifurcated mechanism in the sense that it can either bind CO or the methyl group first. That makes this acetyl intermediate that then binds CoA to make acetyl-CoA. And the acetate again is generated making ATP, and that acetyl-CoA can go on to make all the cell biomass that's needed for the organism. That's the cluster again, and there is an alcove that I'll tell you very briefly about. We've shown there are very large conformational movements that take place within the protein, and you can see that the A cluster here, or you can see the acetyl-CoA synthase is just moving, just like my arm moving back and forth at each catalytic cycle. during the mechanism. So we've trapped the metal carbonyl intermediate using crystallography as well as X-ray absorption on the left, the crystallography on the right, demonstrating that indeed we do have metal carbonyl intermediate shown with very similar sorts of properties based on these studies. We've done a lot of spectroscopy in the past to try to characterize this, but now we have definitive evidence validated by these different methods for the nickel carbonyl. We've shown that there is an alcove that's involved um, in this overall chemistry. So nickel CO binds, and what we located first by this crystal structure and xenon. Actually, first we did it by spectroscopy, but then we're able to locate this xenon molecule that's very close, and thought, is this really important in the mechanism? Because we had been thinking almost purely based on organometallic type mechanisms. And we mutated this phenylalanine 229 to tryptophan. And what we find from that is that if you make an alanine mutation, you completely wreck the ability of the protein to bind CO. Whereas if you take that phenylalanine, convert it to tryptophan, you can make it bind much tighter. So we have a very high degree of tunability here, going from over about 2,000 fold. And what we find surprisingly is that alcove residue, if that is mutated to alanine, we have no autotrophic growth by the Wood-Young-Dahl pathway. So we've been doing studies to try to understand more about the function of this alcove, perhaps mainly to concentrate carbon monoxide near the active site, given our effects on the binding constant. The only other couple of things I wanted to mention is that we have also validated the methyl nickel and the nickel acetyl using X-ray absorption studies. And just recently by some studies in collaboration with Kathy Drennan to locate how the coronoid protein delivers the methyl group to acetyl-CoA synthase. But this tells us, you can see the methyl group here. These are the X-ray absorption. You can see the zanes, the edge, as well as the exafs indicate the methyl group binding. When you bind CO to that methylated nickel intermediate, you make the nickel acetyl. And these are nickel zanes and nickel exafs again. There's very few little changes in the iron exaf, so clearly this is a nickel-based intermediate based on this spectroscopy. So what I've tried to reveal to you is the role of organometallic intermediates in this pathway. The requirement for a tunnel that helps coordinate the movement of carbon monoxide from a COD hydrogenase to acetyl-CoA synthase, and also the role of an alcove that we've just recently found in this pathway, and that its essentiality for the organism to even grow on hydrogen and CO2. So I've been highlighting work that was done by Karima Bulson, as well as the X-ray absorption collaboration with Riti Mukti and Macon, and then XFEL work with Jan Kern, and then long-term work with Kathy Drennan on all these proteins in the Wood-Young-Dahl pathway. So we have future goals. I don't think I'll go through them now, but just take a look at it. Um, I've told you what our aims were. We've reached some of them, but we're still working hard to try to figure out the rest of these questions and others as they arise. So the people who've been involved in this work is a huge group of people, and I'm so grateful for everything that these amazing people are contributing to this. And I thank you for your attention.
Great. Yeah, thank you, Professor Ragsdale, for that amazing talk. We're now open to questions. So if anyone from the audience has questions, please enter them into the box. And we've got one started here from Hannah Shaffet. She asks, I wanted to ask about F512 in ACS. Is that residue as essential as F229 for activity either in vivo or in vitro? And how conserved is the alcove structure across different ACS is?
Yeah, we've published this in a paper. The F229 is absolutely conserved in every organism. The 512 is not completely conserved, but it's functionally conserved, and that there's similar sorts of residues. Most of the residues in that, all the residues in the alcove are highly conserved, but for example, 229, and then one of the isoleucines, there's not a single person in the whole database that has anything other than 229. The 512, there are a couple of substitutions like isoleucine in some cases, but you need to have something there to maintain the integrity around that site and more to keep that cage close to the nickel ion to help to do this. So thanks, Hannah.
See, we have another question from Mala Shabazi. They ask, could you talk about the future of your work more, I believe?
Um, for COD hydrogenase, we're deep into doing trying to understand intermediates in that pathway and understand proton coupled electron transfer. And so we're deeply involved in some stereocrystallography experiments, a lot more spectroscopy to look at that. And what we're developing for both of these projects is looking at crystallography under conditions where we can also look at the spectroscopy and doing things under strictly anaerobic conditions. And that's something that has been missing, I think, in the field, because you go to the beamline, there's no beamline in the world that inherently is anaerobic. So you get exposed to oxygen, the protein dies, and you can see that in some of our studies. You can actually watch color changes taking place right in front of your eyes. But we developed methods to do that, both for cryocrystallography as well as for XFEL. XFEL is stereocrystallography is like a stop flow structure. So you mix substrates with your crystals and you can watch the changes take place. So we're very excited about that work. There's a lot of work going on with neutron diffraction, so we can help look at proton coupled electron transfer. Um, and so for CODH, we are making complexes with the methyltransferase, the cornoid protein, for example, looking at methylation, and again, using crystallographic methods as well as spectroscopic methods to uncover some of the questions that I think are going to help us eventually, maybe have some effects on optimizing this whole pathway, making it even better, making it more efficient for biotech uses as well as perhaps for mitigating climate change.
Excellent. Thank you. I think we have time for maybe one more question. This is from Paulo Laude Velasco. They ask, is it possible to isolate the inorganic clusters from the organic components? And can you explain briefly how the XAFs and ZANES measurements were made? And probably fairly brief for the experimental technique.
Okay, yeah. Um, so yeah, okay, just one more time for the first part of that, for the first question.
Is it possible to isolate the inorganic
Oh,
clusters
right.
from the organic
No,
components?
it's not. Well, I mean, maybe someone could do that, but the thing is, they're so tightly coordinated. If when you look at the structure, you see the cysteine residues are linked to the protein. So every iron is linked directly to the protein. And so you might find ways to lift it out. Um, any kind of changes we make, we seem to see the cluster degraded. So it's something I think probably you're better off trying to capture the ideas, the concepts that the protein has sort of hinted to us about the structures. Um, it's just not going to be easy to lift it out from the protein. So I'm not going to say it's impossible, but I think it's going to be extremely difficult. And the X-ray absorption studies were done. We've done some in crystallo, and everything is done within an anaerobic chamber. So our crystals are transferred, or our solutions are transferred into specific types of tubes that then go under vacuum, and they're kept under anaerobic conditions within the X-ray absorption within the X-ray absorption system at SSRL and Riti Mukti Sarangi's laboratory.
Awesome, thank you. So I think that probably brings us at time for Stephen Ragsdale's portion of the talk. Thank you so much for your presentation and a very active question and answer session. Just a note that we will be sending up a follow-up email after the event, and maybe we'll answer some of these questions through that. I also encourage you to reach out to Professor Ragsdale for more discussion on a fascinating topic. So thank
Yeah,
you once again.
absolutely. Thank you.
So we will now transition to our next speaker. But first, I'd like to take a quick opportunity to share about our multidisciplinary journals, including our flagship Chemical Science, which is always free to publish in and open access, and publishes a lot of work in inorganic chemistry. Right now, there's a themed collection on bioinorganic chemistry, guest edited by some of the leading researchers in the field. I encourage you to click that QR code, read some of the work that's in that curated collection, and see more what that's similar to the webinar today. And with that, it's my pleasure to introduce the next speaker, Professor Patrick Holland. He's the Whitehead Professor of Chemistry at Yale University. His work focuses on the chemistry of inorganic complexes, their reactivity, and their mechanisms. He recently published in Chemical Science on iron alkyl complexes for nitrogen splitting and actually participated in our 2023 Faraday discussion in London on that topic. Today, he'll tell us about catalytic reduction of CO2 and how nickel-containing iron-sulfur clusters are relevant to carbon monoxide dehydrogenase active sites. And with that, I'll hand off to Professor Holland. Thank you.
All right. Is this looking okay?
Yep, looks great.
Excellent. Thank you so much for making me part of this series, and especially for the opportunity to talk right after Steve about CODH, because this enzyme system has been inspirational to me and to my group over time as an example of iron sulfur clusters that do really interesting reactions, and of a case where bioinorganic chemistry teaches us how to do chemistry better. My background is as a synthetic chemist, interested in small molecule activation. As you heard, a lot of our interest is in nitrogen activation, but we've more recently gotten into this topic because there are so many interesting questions. Luckily, I don't really need to introduce the enzyme to you because Steve just did that for us. And I'll just launch straight into my thoughts on the structural model for the C cluster. This is the cluster that you just heard about that interconverts carbon monoxide and carbon dioxide. Here are three different pictures from the same structure. This is the electron density picture. Here is a ball and stick picture, and then here's a ChemDraw picture that gives you an idea of what that structural model looks like. And really what jumps out to us is this unusual nickel site that me as a coordination chemist and organometallic chemist, it looks really strange to me. And it makes me start thinking about fundamental questions. One is, how do you get a nickel into an iron sulfur cluster? And we'll come to that a little bit later in the talk. Another thing that's really unusual is that in the definitive paper on the crystallography in this from 2007, the picture that you see here is nearly indistinguishable between C-Red 1 and C-Red 2. These are two reduced forms of the C cluster that differ by 2 electrons, and that's been shown by Steve and others quite a while ago. But the structures being indistinguishable despite being 2 electron different is a very strange phenomenon that requires more thinking. And the other thing about the model that's strange to us is that this nickel has only 3 bonds to it. That's rarely found in nickel compounds, and strange to find in the middle of a biological cofactor. Also, when nickel 61 substitution was used, there's very little hyperfine coupling to that nickel, indicating that there's not much spin density on the nickel. And that's strange because it's part of an iron sulfur cluster. And the rationale for that has been what Steve showed you in his slide and what I'm about to show you, the idea that this would be nickel 0 or nickel 2 in even oxidation states, and explaining why the spin density is so low. But we thought that that is unusual, especially given that nickel 0 is an extremely low oxidation state to find in an enzyme. So here's a picture very much like the one that you saw in the previous talk. The structural picture of C-Red1 and C-Red2 is that they differ by 2 electrons. I think from the talk, it looks like Steve's group has a little more detail on these now that I'm looking forward to learning more about. And I'm going to actually present it in the opposite direction of what Steve did. The enzyme goes both directions. And just for completeness, maybe I'll go in the clockwise direction, where you could imagine the nickel becoming reduced. And then if you have a low coordinate nickel 0 site, even though that's strange to find in there, that would be consistent with it binding CO2 to make this metallocarboxylate that would be assisted by the Lewis acidic iron over here. This is backed up by an X-ray crystal structure that shows the bridging CO2, which you saw in the previous talk. Then, if that were protonated at the oxygen, you could split the CO bond, make a transient CO complex, which could then lose the CO and get you back to where you started. I will note that seeing bent CO is very strange. And in this review, we speculated that perhaps this is a superoxide that's actually bound to the nickel in the cluster, but misidentified as CO. But that remains to be seen. So overall, though, the point that I would like to make is that there are really a lot of coordination chemistry questions to ask about these kinds of cofactors. And my group and I are synthetic chemists, and we try to think of how we can contribute to this area. And what we find is that systems like this, or like nitrogenase or other metalloenzymes, set us up with really interesting questions to try to answer. One would be when you have a hetero metal like this in a iron sulfur cluster, how would it behave? What are feasible oxidation states and spin states that you might observe? Also, is this 3-coordinate geometry in nickel, is that actually feasible? This would be the first example of a crystallographically characterized 3-coordinate nickel in a metallobiochemistry. And also, where do small molecules react? Would it react at the iron or at the nickel if you were to have a compound like this? I think this actually relates to the question that came up after the previous talk as well. If you could pull this out of the enzyme, how would it behave, and can we try to back that up? So that's what my group tries to do by making compounds that look something like the cofactor. They're of course informed by the enzyme, but what we hope is that we can determine spectroscopic, structural, and other characteristics of this type of metal site, which can then help to inform the way that we think about the enzyme. This is synthetic modeling chemistry. Now, for CO dehydrogenase, people have thought about modeling this before. Interestingly, some of the first models came out even before the structure of the enzyme. These came from Richard Holm and his group, who designed this really interesting 3-iron, 4-sulfur crown-shaped series of clusters where the irons are held in place by this large multi-nucleating ligand that you can see in this chem draw, and you can see it in the ball and stick figure here. It's ideally set up to put another metal in here, and one of the metals that they inserted into it was nickel. And they used nickel 0 sources, which were oxidized in the process of this. They didn't really know the oxidation state of nickel, but speculated that it was nickel 2 in these systems, which involves the loss of an electron to we don't know where. And that made a tetrahedral nickel site. And you could substitute that with bidentate phosphines, and that would make a square planar site. But I want to call to your attention that in both of these cases, the nickel is 4-coordinate. And so we didn't have a test of whether 3-coordinate nickel is feasible in systems like this. So that's where things were. And the people in my group that decided to try to reinvestigate the small molecule chemistry as it relates to this were Dan Wilson and Majid Fatafta, who were simultaneously postdocs in my group and decided to try to jump into this and see what they could find out. They came up with two different strategies that I'll go through one by one. The first strategy was to put another heterometal in there, which might seem a little bit counterintuitive. But what's nice about iron, sulfur, tungsten, or molybdenum clusters is that by adding the group 6 metal in here, it stabilizes the cluster a lot. And so they figured that they would be able to isolate and study these kinds of compounds. Then this is ideally set up with 3 sulfurs here in order to bind a nickel. And the one that Dan and Majid chose was one with IPR, which is a bulky and heterocyclic carbene ligand. And that binds very well to nickel 0 in this nickel 0 precursor. And what they found was that the cluster here was capable of binding nickel, and we've already answered one of our questions. We see that the nickel is 3-coordinate, so that's actually feasible in this C cluster kind of structure here, that it can let go of one of the sulfurs and still be stable. We did a number of different spectroscopic and magnetic studies on this, but I'm going to cut to the chase and talk about collaborative work with Serena De Beers' group, in particular, graduate student Zach Matthey, who did X-ray absorption spectroscopy. And we're looking here at the K edge, which means you're promoting one of the 1s electrons into the continuum. And if you look at the pre-edge, that is the Zane's part of the spectrum, then you're looking at features that are 1s promotions into valence orbitals. And the iron edge suggests that there's very little change here and in the edge, and that the irons haven't really changed oxidation state. But what's important in the nickel edge is in the spectrum itself and in the derivative of the spectrum that I show underneath it, you can see this pre-edge feature around 8330 electron volts. And that's characteristic of this pre-edge. And if you see that one of the electrons is being promoted into the 3D level, that means that you have to have a space in your 3D level to put that electron. And that's inconsistent with nickel 0, and that indicates that we have to have nickel 1 or nickel 2. So based on that, we can rule out nickel 0 is the best description of these complexes. This was supplemented by calculations, also done by Zach, that indicate that in this nickel 1 compound, there is a nickel tungsten bond. We have one of the up electrons and one of the down electrons that are co-localized. And you can see it's very clearly a sigma bond between these two metals. And that's pretty interesting. What that indicates is that you can have a nickel 1 site that actually doesn't have much spin density on it, despite the fact that normally when we think of a D9 metal, we think that it's going to have spin density. Instead, this has a metal-metal bond, and so it can be nickel 1, yet without spin density on it. So our overall picture that we end up with of this interesting cluster here is that we have 2 iron sites. I sort of skipped over the data that support this, but these would be a mixed valence pair and have 1 delocalized electron between them. And then the tungsten and nickel sites would have a metal-metal bond. And this enables the nickel basically to store an extra electron in the form of a metal-metal bond. And so those are our morals of the story of the first part of my talk, that three-coordinate nickel seems to be feasible. Nickel zero does not seem to be feasible, at least under these conditions. And you can have a small spin density at a nickel one site, even though that might not have been previously considered, but it's because there's a metal-metal bond in which that formally unpaired electron is actually shared. The biggest problem with the studies so far, though, is that the metal-metal bond that's formed here is from nickel to a tungsten, and the tungsten is the non-biological part of our cluster. We just added it for synthetic utility, and now we discover that it dominates the electronic structure of the cluster. So we moved on to other clusters, which don't have tungsten in them. And of course, Holm made some without tungsten in them, and I showed you those before. And those were based on the crown structure and then putting the nickel in another corner. It's kind of interesting that that's not the way that nature does it. The way that natural systems are thought to put nickel into clusters, they start from a 4 iron 4 sulfur cubane, which then they break open and cram a nickel into the middle of it. That is pretty amazing because in both synthetic and biological chemistry, we normally think of iron 4 sulfur 4 cubanes as the bricks of iron sulfur chemistry. And somehow this biological biosynthesis system has a way of breaking it up. And Magid thought, because he's pretty tough, he thought maybe he could do that himself. And the strategy that he took was to use one of these amide coordinated clusters. These have been previously synthesized by Sunny Lee's group and also from Yasuhiro Oki and Kaz Tatsumi. They had made this cluster. It's very oxidized. And what Magid thought is that maybe by adding a very reduced nickel zero center, that would be able to drive the insertion of the nickel into the cluster. And the product that he isolated in good yield is actually pretty amazing. Not only did he really chop up the iron cluster, he also stuck 2 nickels into it. And now there are 5 metals, 4 of which have only 3 bonds to them. So we really added a lot of muscle to this reaction and made a very cool cluster. Here are 2 views of it. Here's the top view. And the side view, you can see that all 5 of these metals are in the same plane. Now here we wanted to understand the oxidation states as well, and I'm going to skip a lot of the details, but skip straight to the Zanes again. And for this cluster, we again see the pre-edge feature around 8330. The calculations complement this and show that there is that the electron is going into a nickel orbital, indicating that this is not nickel zero. This is best described as nickel one again. There's not much spin density on the nickel, and that there is some iron nickel bonding. Not as strong as in the tungsten case, but there is some bonding there. And so the way that we assign the oxidation states in that system is that we have 2 nickel ones here, and then an iron 3 in the center, which is antiferromagnetically coupled to 2 iron 2s and to those 2 nickel ones. And that gives you the overall spin state of 5 halves, which is what we observe using magnetometry and EPR spectroscopy. Okay, on to other clusters. Magid also tried doing this same kind of reaction with a reducing agent at the same time and isolated a different cluster. This one has the iron sulfur cubane still intact, and the nickel is on the outside. Here's a picture of the structure of that. And figuring out the oxidation states of this were actually easier because the Nussbauer spectrum, the signal there matches up extremely well with a 4 iron 4 sulfur cluster previously synthesized, where the isomer shift is basically identical. And that was for 2 iron 3, 2 iron 2. And so we think that's the same for our cluster. and just by process of elimination, that means that this is also nickel 1. If we look at this cluster's zanes, that is the one that's labeled iron 4 nickel here in red. You see that again, it has that same pre-edge feature. And if you're an XAS aficionado and you're saying, well, those pre-edge features didn't have to be to a 3D orbital, they could be to one of the 4s or 4p orbitals, it could still be nickel 0. Here's a bona fide nickel 0 compound. It's our starting material in these reactions. And you see that that one doesn't have the pre-edge feature. So we see that as a good control experiment. Also, we can compare this to X-ray absorption spectroscopy done on the enzyme itself. We see a pre-edge feature in that same area, which might be relevant, or it might just be a coincidence. We don't know. In this case, where the nickel is external to an iron sulfur cluster, again, you do see some signs that iron nickel bonding is taking place. It's much weaker than in that tungsten case, but you can see a little bit of interaction there. Okay, so the emerging theme here seems to be that the nickel prefers to be in the nickel 1 form. Now, more recently, Magid and now current student Teddy Gerard have been studying basically the same kind of reaction, only with an oxidizing agent added. In this case, you get another interesting result. Now the nickel is in the cluster, and one of the irons has been popped out to be outside of the cluster. Very similar to the previous structure, it's just that nickel's in and iron's out instead of iron in and nickel out. And actually, it turns out that just by adding oxidizing or reducing agents, you can interconvert these two forms. And Teddy has been studying the interconversion of these two. I think it's really interesting how labile these clusters are, which is something that has been really profitably studied recently by Dan Zues and coworkers. And also, I think that this could be helping us to understand something about how nickel is inserted using redox in the biosynthesis of these. At the very last minute here, I want to tell you about some of the more recent studies that Teddy has been doing, where he's been adding inhibitors of the enzyme. And what we find is that although isocyanide here had a choice between a nickel site and a 3-coordinate iron site, it actually chooses to bind to the nickel. And here's an X-ray crystal structure that shows that. Teddy's gone on to study this in some simpler clusters that don't have the dangling iron on these, and he finds that T-butyl isocyanide also will bind to this at the nickel site. And more excitingly, he's done this with cyanide, which is an inhibitor of the enzyme, and finds that that similarly binds to the nickel to give a square planar site. That can be compared to the structure, Steve showed this during his talk. There's a crystal structure of the enzyme with cyanide bound. We have this compound with cyanide bound, and the metrical and spectroscopic parameters are extremely similar between these two. So that's a nice validation of that enzyme crystal structure. Then more recently, we've been studying this by Endor spectroscopy in collaboration with Alex Drana, who is working in Brian Hoffman's group. And we see Endor spectra of this, where we see a very clear signal for C13 labeled cyanide that matches up very well with the enzyme as well. So what we're hoping is that in the long run, these kinds of compounds can help us with validation of what structural features correlate with spectroscopic features in the enzyme. And obviously, one of the next things that we want to do is try to get CO and CO2 to bind to these clusters in order to model the system. So I can see that I'm slightly over time here, so I'm not going to take you through these conclusions one by one, since I've told you about them before. But I hope that I've given you the idea that we've found ways to synthesize these iron nickel sulfur clusters in particular, ones where we have a cube and a dangling metal off of them, which were inspired by the enzyme. We're now finding ways to bind things to them, looking in spectroscopic and structural detail, and having a lot of fun doing it. Finally, I want to thank all the people who make this happen. Our entire group contributes to all of our chemistry through wonderful interactions. But particularly, I want to thank Teddy as well as Sam Newman Stonebreaker, current postdoc in the group, who's been looking at the dependence of the properties on the terminal ligands on these clusters. I want to thank Dan and Majid again, all of our collaborators, the NIH for funding on our side, and you for your attention.
Excellent. Thank you, Dr. Holland, for a fantastic talk. And we've got some questions in the Q&A box. So first, from William Jones, if the nickel 0 is actually nickel 1 with a metal-metal bond, then there's no empty orbital to produce the pre-edge band, since it's filled with the bonding pair of electrons. How does this work?
Oh, right. Yeah, that's a great point. This is a very weak bond, so I would say that it's got some sigma star character to it, right? So you still do have that. You still do have a place for those electrons, and because it's a weak bond, the energy is pretty similar to what you would expect for the d orbital. So that's our explanation for now. And I will also mention, thanks for the question, Bill. That all these pictures of integer oxidation states are all oversimplifications of the electronic structure of this complicated cluster. That once you start looking in detail at these, all the orbitals are all mixed together. So what I'm giving you is sort of a distilled down and somewhat oversimplified version.
Great. And another question from Liviu Mirka. Hi, Pat. Great talk. I was wondering if you can please comment in more detail about the reactivity of your nickel heterometallic clusters with carbon monoxide or CO2 or other substrate analogs.
Sure. Of course, this is something that we're very, very interested in. The last series of structures that I showed you have these amide terminal ligands on them, which are really great for stabilizing the clusters. However, as many people may know, amide ligands can react with CO2 very readily. And so those systems are not ideal for looking at CO2 reactivity because we'll have, reactivity on the outside. So one of the things that we're doing is putting thiolate ligands on these or other ligands that are going to be more stable under reaction conditions. That's been a problem so far. And the other thing that we're battling with is the carbon monoxide has a way of ripping the nickel completely out of the cluster, which is something that maybe doesn't happen biologically quite as fast because it's not getting a whole lot of CO all at once. But I think that these are manageable problems, and that's what we're going to try to do in the next generation of chemistry on these clusters.
All right, and let's have one more question from Neil Mancad. Hi, Pat, great talk. Do you have any thoughts about the CN stretching frequencies in the isocyanide increasing upon coordination to nickel?
Sure. Yeah, this is actually something that people have seen quite a bit. That you would think that they would go down because of back bonding, but first of all, these nickel's not the greatest back bonder in the world, and and so we think that the system's not back bonding very effectively. And actually, when it comes to thinking about the amount of back bonding, we're in the process of thinking about this, but I'll refer you to some recent really nice work from Hannah Shafat's group, where they've been thinking about this. I meant to mention that earlier in the talk, and I think I skipped over it. But we're certainly thinking about this, but I think the back bonding is not that important. And if you're thinking about the function of this enzyme, you don't want there to be great back bonding that would make your CO bind tightly, right? If you're going in the CO oxidation direction, you don't want it to bind too tightly because you want to be able to get to the transition state for CO bond formation. Or if you're thinking about the CO2 reduction direction, then your CO is a product and you don't want it to be binding too tightly. So I think there might be something inherent to the way that this system has evolved, but this is something that we're still in the process of looking at. Thanks for the question, Neil.
Awesome. In the interest of time, we'll need to stop there, but thank you again, Professor Holland.
Great. Thanks.
Okay, so just to wrap up, this does bring us to the end of our webinar. So, please keep an eye out for a follow-up email. If you have any questions that you'd like to send to the professors or both of the speakers today, they've included their email addresses here and are available to answer some of them. Thank you all for attending, and thank you again to our speakers, Professor Stephen Ragsdale and Professor Patrick Holland. We hope to see you all at a future desktop seminar. You can find the list here at this QR code, and in fact, we have one next week on materials chemistry for wearable sensors. I'll end on a call to action. If you're interested in helping us improve publishing and our services, please sign up for the RSC Digital Research Lab. Again, you can follow this QR code to form to sign up for this. And with that, I'd like to thank you all for attending and wish you all a good day, evening, or morning, supposing wherever you're at. Farewell.